In this session, we'll be talking about another type of recessive epistasis called the duplicative recessive epistasis, and they are also referred to as complementary genes in some cases. Now, let's look into this. In this case, if you're going to cross two varieties of pea which are being considered to be what white in color, that is, the flowers of these pea varieties have been found to be white, and the pea varieties of which bear white flowers can occur in two different forms, either as capital letter C, small letter p, or small letter c, capital letter P, and capital letter P. These two varieties, these pure breeding varieties, are being crossed together, and we can see that you get always a hybrid in the F1 which will bear purple color flowers. That is, they will have a genotype of capital letter C, small letter c, capital letter P, as well as small letter p. And all the flowers which have been born in the F1 generation will be found to be having a purple color, unlike the parents which were being considered to be which were white flowers, which were bearing white flowers. And when the F1s are being crossed against each other to get the F2 generation, you can see that different gametes will be formed, and as a result we can see that there would be both purple in some of them will be bearing purple flowers, and some of them will be bearing white flowers, and the ratio of the purple flowers to the white flowers is considered to be as 9 is to 7. So one thing which we note over here is that the more of the hybrid characters, okay, is being expressed in the F2 generation than the parent characters. Now let's just explore what are the genotypes of this. In the case of the purple varieties, you are the ones which we have here. We are having capital letter C, capital letter C, capital letter P, capital letter P. In the next one, we have capital letter C, double capital letter C, and small letter p, as well as a capital letter P. And in all these purple varieties, we can see either capital letter C is there or either capital letter P is there. Okay, and in the white varieties, if you have a close look, you can see that uh, it will be either capital letter C, capital letter C, or in the next non-allelic P version, there is no dominant. Here also, if you take one allele, okay, sorry, one non-allelic gene, that is C here, and its other non-allelic gene P, you can see that capital letter P is not being present. So here, if in this one, you can see the C is recessive, but here P is dominant. And in other white also, you can see here the C is recessive and the P is dominant, and the vice versa over here. So in all these cases, what happens in these 1, 2, 3, 4, 5, yeah, 3, 4, 5, 6. In 6 of these varieties, you can see that one of this, either C will be capital or P will be capital. And in the final one, you can see there is one version also over here where small letter C, and small letter P is also being present over here. So this is also a recessive type of one which is being present here. So here, if you go to look, there is a complete dominance at both the gene pairs. That is, when C and capital letter P is being present over here, they have a complete dominance, and they are the ones which are responsible for the formation of the purple color. But when either of the gene is homozygous recessive, it is hiding the effect of the other gene. Okay, when capital letter C and capital letter P is there, we are having purple color. But if either C or P, if they are being present in a homozygous recessive condition, that will inhibit the formation of the what? The dominant, the violet color here, which is being present. So here, duplicate. So here you have small letter C, small letter C. Here you have small letter c, small letter c. In this white, you have small letter p, small letter p. So in all these cases, when either cc or small letters pp is being present, it is inhibiting the formation of a what? purple color. So the homozygous recessive is hiding the effect of the other gene, and it is a case of a what? Recessive epistasis. And here, because you can see it's a homozygous recessive gene, so it's a duplicate recessive epistasis. And there is a ratio of 9:7, which is over here. Now, we have considered this as a complementary gene. So we will just explore what is the molecular mechanism behind this, why sometimes you get white color and why you get purple color. If you go to look into the genes which have been responsible for this purple color, the purple color of these flowers is mainly due to the presence of a pigment called anthocyanin. And this anthocyanin is mainly being formed by a two-step production. Normally, by the action of the first gene product, capital letter C, a precursor molecule is converted into another precursor 2 molecule. Okay, so here by the action of C, this precursor is going a step 1 and it is producing a particular precursor 1 over here. And after that, by the action of capital letter P, what will happen? The product which has been formed here as a result of step 1 is being converted into the step 2 compound. It will undergo a step 2 and it will produce anthocyanin. So, the violet color is being formed here only when both capital letter C as well as capital letter P is being formed. So the production of anthocyanin, as we mentioned over here, mainly requires both the genes capital letter C as well as capital letter B to act together. Okay, and so to completely get anthocyanin, you need the action of both of them together. If either of the capital letter C or capital letter P is not there, the anthocyanin production is not possible. So if either of the step is non-functional, then no purple pigment is produced and the affected pea plant will bear only white flowers. The dominant C and P alleles code for the functional steps in the anthocyanin production, and the recessive C as well as P alleles code for the non-functional steps. So if there are two recessive alleles occur for either of the gene, the white flower will result. So here you can look, if any of these genes are being found in the homozygous recessive condition, it will inhibit the effect of the other gene. Here capital letter P is there, but the small letter C and small letter C, the homozygous recessive C variant, will inhibit the action of the capital letter P, small letter p here. So that is a case in which a recessive epistasis is been happening. That is, a homozygous recessive allele is inhibiting or controlling the effect of a what? An other allele or a non-allelic gene. Now, let's explain what are complementary genes. Complementary genes are defined as two or more dominant genes occurring in a different loci of the same chromosome or a different chromosome, which interact with one another to produce a single trait, but neither of them, if present alone, produces a character in the absence of the other. In the case of supplementary gene, we had noticed that even if the other one is present or not, one type of the trait will be formed, but it would be different. But here, the trait will not be produced if both of them are not there. If either one of them are absent, what happens? The absence of that particular character is being, or if either of these two interacting genes which have been found in the dominant genes, okay, they should be dominant in nature. If they are not being found in the dominant pattern, what happens? The effect will not be resulted. So the violet color will come only if C and P are both of them considered to be in capital letter or in dominant forms. In other words, the action of these independent genes are considered to be as complementary. In the example of the pea plant, you can see that the violet color of the pea flower is due to the complementary action of the dominant alleles of both the genes responsible for anthocyanin production, and only when both of them are being found in the dominant form, the anthocyanin production will be formed. In other words, when either of C and P are found together, the violet color of the flowers are formed. And therefore, C and P, that is the dominant versions of capital letter C and capital letter P, are being considered to be as complementary genes. I hope this is clear to you. So we came across a case of recessive epistasis again here, but here the genes are being found to be complementary to each other. The complementary means only if both of them have been found together in the dominant form, the resultant trait will be coming. That is, the violet color here is being formed. Now, in the case of supplementary genes, we have another a different case where they are two independent pair of genes and they interact in such a way that one dominant produces its effect whether the other one is present or not, but the second one produces its effect only in the presence of the other. So we had mentioned over here, the capital letter C will produce its effect over here, whether or not small letter b will be there, but the effect will be different over here. Okay, and if you go to look here, okay, the capital letter B also is being present here, but it will have a different effect. Okay, so in this case, the violet color, in the case of complementary genes, the trait is being produced over here only if both the genes are being found to be in a dominant phase and only if they interact with each other. I hope this is clear to you, and we'll be continuing with different other types of epistasis in the coming sessions. Thank you for now.